So the osmosis is the process of flow of water from higher water concentration to lower water concentration. So now there's different kinds of environment, that is isotonic, hypertonic and hypotonic. Okay, in isotonic environment both water and solute concentration is same inside and outside of the cell. So what is the solute and solvent? Like for example, if you are dissolving NaCl in solvent water, then NaCl is nothing but the solute and solvent is nothing but water. So when water and solute concentration or NaCl or any other salt is same outside and inside the cell, then such a kind of environment is called iso. Iso means same. Isotonic environment or isotonic solution. When bacteria is present in isotonic solution, within the bacterial cell the solute concentration is same, and outside the liquid also, outside the bacteria also, the solute concentration is same. Okay, so water flows from here to here, but it is at equal rate. Okay, if 10 ml is coming into the body, then 10 ml is going out of the body. Such is the condition. Okay, then next is the hypertonic solution, that is the water concentration is greater inside the cell while the solute concentration is higher outside. Hypertonic, hyper means greater, maximum. So the outside solute concentration is very high, like when the bacteria is present in the salt solution, that is it totally has salt, whereas within the bacterial body there is no such amount of salt. Such condition is called as hypertonic solution. In this condition water goes out of the cell, that is water flows from higher water concentration to lower water concentration, or you can say from lower solute concentration to higher solute concentration, that is nothing but osmosis. So when you place a cell, any cell, bacterial cell or animal cell, any kind of fungal cell or any cell, if you place it in a hypertonic solution, the water goes from cell to outside. That is called degradation, or that is the way you break some cells or you make the cells to die. When you place a cell in the hypertonic solution, all the water within the cell, it goes from inside to outside and the cell becomes flattened, or in other ways finally it dies. So this is hypertonic environment. And the other opposite kind of example is hypotonic condition. In this, the water concentration is greater outside the cell and the solute concentration is higher inside. So all this hyper, hypo, we are telling whatever it is outside. Hypertonic means the outside solution is hyper. Hypotonic means the outside solution is hypo. Hypo means less, hyper means greater. So when you say hypotonic, the outside solute is very less. So water flows from low solute to high solute. So it comes from outside to inside the cell. So what happens when the water comes into the cell? Whole water, out of 100 ml, 90 ml is coming into the body and only 10 ml is going outside. Then what happens? The body bulges and finally it breaks. Okay. So that is how these kind of environments are used for when you are isolating some cells, or when you are isolating DNA from the bacterial cell or from the animal cell or any other cell. This is how you disrupt the cell membrane and you get the contents of the cell outside. Okay. So most of the bacteria, they require isotonic environment or the hypotonic environment. Okay, that hypotonic environment too at particular range only, not like very less solute concentration. In that case the cell bulges and it breaks off. So like this way they either grow, and most of them they require only isotonic environment, some they require hypotonic concentration. Okay. So organisms that can grow at relatively high concentration, high salt concentration, that is up to 10%. Remember this range, up to 10%. Any cell cannot grow in highly concentrated or highly hypertonic environment. The range is only up to 10%. The organisms which can grow at 10% salt concentration, they are said to be called as osmotolerant. Okay, so these require higher salt concentrations of 10% or 20%. Okay, sodium salt is nothing but sodium chloride. These are called as osmotolerant organisms or halophiles. Halo meaning salt. Okay, philes means organisms which like halo. Okay, halophiles. So these are all about the physical requirements. Now next, moving on to the nutritional requirements. So bacteria also require nutrition. Without the media, without the culture media, and without the nutrients in it, any bacteria cannot grow in the medium. So under the culture media, I have explained you different kinds of media and have uploaded the notes also. So you can mention a note on all these culture media, defined media, complex media, enriched media, or differential media. You can give a note on that. But basically the media consists nothing but energy source, a carbon source, nitrogen source, and then you require vitamins or growth factors, micro elements and macro elements. So this is the list of all that energy source. So energy source meaning we have seen this in nutritional types of bacteria, if you can remember. Girls, it's been long days, I don't think you remember. So just remember, just try to remember, okay, our class of nutritional requirements of bacteria. So phototrophs, chemoautotrophs, phototrophs and chemotrophs. Okay, phototrophs means which use the energy source, that is sunlight as an energy source, they are called as phototrophs. And chemotrophs which use the chemical energy of the chemical compounds, that is by oxidation or reduction of the chemical compounds, they are called as chemotrophs. Then carbon source, the carbon source, based on this bacteria they are divided as autotrophs and heterotrophs. So autotrophs, they require only carbon dioxide, and heterotrophs, they require organic forms of carbon. They cannot synthesize or they cannot derive energy from the carbon dioxide. Heterotrophs, they require organic molecules, and then they can utilize those carbon organic forms and then they can grow. Okay, they cannot utilize inorganic nutrients. Okay, so based on this we have seen different nutritional kinds of bacteria: photoautotrophs, photoheterotrophs, chemolithoautotrophs, chemoorganoheterotrophs. Okay, so the examples for this also I have given you. Okay, so next source is nitrogen source. Nitrogen, under macro elements we get this. Okay, carbon, oxygen, nitrogen. So nitrogen is required for the synthesis of amino acids, DNA, RNA, ATP. It is present everywhere. There are some microorganisms which can fix the nitrogen. So they are called as nitrifying bacteria. We have even discussed on this. Okay, so they can use either ammonia, they can use either nitrates, they can use organic nitrogen, anything as a nitrogen source. That is one of the requirement for the bacterial growth. Then coming to the other minerals or other micro elements like sulfur, amino acids, there are some sulfur containing amino acids and certain vitamins which require sulfur. And also phosphorus is required for DNA, RNA and ATP. And then potassium, magnesium, calcium, iron, these are all called as macro elements which are required for the growth of the bacteria. Then coming to the trace elements, that is micro elements. So what are these micro elements? Potassium, magnesium, calcium, iron, these are all required in minute quantities and they usually function as cofactors in enzyme reactions.